I was saying, Novogene was established in 2011, and since then, we have grown immensely. We currently operate 10 state-of-the-art laboratories and sequencing facilities worldwide, serving over 90 countries across 6 continents, which means wherever your location, we are here to back your research with local support and global expertise. Now, zooming on into Europe, we have 2 labs, one in Cambridge, UK, and another in Germany, in Munich. The one in the Cambridge in UK is right in the heart of one of the most vibrant science clusters, and another in Munich, located in an innovation-rich biotech hub. These labs give us the speed and flexibility to support your work closely and efficiently. We are powered by the latest technologies on the market, and our commitment to cutting-edge science is reflected in our continuous investment in the latest technology platforms to ensure our researchers have access to the most up-to-date tools available. For example, we recently added the AVITI24 sequencing system from Element Biosciences to our ever-growing portfolio of sequencers. In addition, our single-cell transcriptomic services are powered by Chromium iX system from 10x Genomics, enabling high-resolution analysis at scale. So what does Novogene do? We are your one-stop multi-omics service provider. We offer everything from next-generation sequencing to data analysis, starting from research in oncology to all the way to food and water tasting. And here you can see our comprehensive service portfolio for different applications, for example, for genomics, transcriptomics, microbial genomics, epigenomics, and also proteomics powered by Olink portfolio. For our whole plasmid sequencing, which is our newest service addition, we now are able to sequence whole plasmids powered by Oxford Nanopore technology. And today's seminar will focus on single-cell RNA sequencing service, which is one of the newest additions to our transcriptomic service portfolio. Here is a brief overview of our single-cell transcriptomic service. We offer single nuclei and single-cell RNA sequencing for a wide variety of sample types. Very recently, we have also now started accepting whole blood and bone marrow. And we have also expanded our service portfolio to include 5' gene expression designed for immune profiling. Since the time we have launched, we have gathered experience in a wide variety of sample types, not only limited to human and mouse, but for a variety of other species that you can see listed here. We also have experience with different types of organoids and tissue types for these different organisms. Before I hand over to our speakers, I just want to take a moment to get your input on whether you are currently running any kind of single-cell sequencing experiments in your lab or with any other service provider, and whether there are certain steps in these experiments that you suffer from. And Miranda, do you mind sharing the poll?
Sorry, Sony. Yes, I will share it in a minute. There's some setting behind here. Thank you. Sorry.
Yeah, no worries.
I think we can continue for a bit while I'm finding the setting right now. Sorry for
no worries.
everyone is attending.
Okay, maybe we can run it at the end of the session then. Then I will hand over to our guest speakers, Dr. Nico Dunkel and Dr. Martin Seifert, so you can hear more details about the technology behind this service. A brief introduction to our speakers for today. Dr. Nico Dunkel joined 10x Genomics back in May 2020 and is now responsible for all biopharma accounts in DACH, that is Germany, Austria, and Switzerland. Prior to joining 10x Genomics, he held commercial positions at Stila Technologies and Takara Bio Europe. He holds a PhD in micro and molecular biology from the University of Würzburg. Our second speaker for today's webinar is Dr. Martin Seifert. He is the Senior Science and Technology Advisor at 10x Genomics. He began his career studying gene expression in zebrafish at Technical University Munich, followed by postdoctoral research at Brunel University London. His tenure at Genomics established him as a data analysis expert focused on bioinformatics solutions in life sciences. Before joining 10x Genomics, he founded Connexum, a consulting firm assisting customers with experimental planning and multimodal data strategies. His research interests focus on integrating single cell transcriptomics and spatial genomics technologies with other data modalities. He has co-authored over more than 50 peer-reviewed publications in genomics, epigenetics, and transcriptomics, combining molecular data classes for systems biological applications. And today's seminar will give you an insight into the technology behind the single cell transcriptomics portfolio that we offer, as well as how you can analyze the data that you obtain from these experiments. You can now see the poll pop up. This will help us understand what is it that is stopping you from starting your first single cell RNA sequencing experiment and what are the pain points that you suffer from. Do you see the polls coming in, Miranda? From my end, I don't see it.
Yes, we currently have a lot of people participating. It's very popular, seems. I think we will leave this until at the end of the webinar so we can have a overview how you at the end how the option is. Thank you guys for participating right now. It looks really popular now. Yeah, very good
All right. Without further ado, I will hand over the stage to our first speaker for today, Dr. Nico Dunkel. Over to you, Nico.
Hey, Sonny. Many thanks. Hey, Miranda. Many thank you. Thanks for that. And I share my screen now. Share. Hope you can see it. Sonny, can
I
you hear me?
can. Yes, yes,
Okay,
yes, I can.
perfect.
We are good to go.
So many thanks, Sonny, for this great introduction and also for the chance to for us to give a talk here today. And welcome everyone attending online here for taking the time and logging in. And yeah, it's a great pleasure to speak with you today. And as Sonny mentioned, I will give you a brief introduction on how single cell basically came to be, what we offer from 10x Genomics, how we from 10x Genomics and also Novogene can support you. And then I will hand over to my dear colleague Martin, who will educate a bit on the bioinformatics and data analysis part. Let me start with a fact that I hope we can all agree
Nico,
about.
sorry to interrupt. We see part of the screen blocked by this focus period. You probably want
Oh,
to
okay.
minimize.
Yeah. So obviously I didn't want it to be distracted by any email popping up.
Makes sense.
Okay.
Okay. Now we
Okay.
see your full screen.
Very fine. Thank you, Sonny. Yeah, so I guess we can all agree on the fact that biology is immensely complex, and just as an example, the human body alone is made out of 40 trillion cells, and the bone marrow produces 500 billion cells each and every single single day. So it is a lot of cells that our human body is made of, and if we set out in our research and we as technology providers set out in our tool development, if we want to improve human health and cure diseases and do want to understand why some patient may respond to a treatment or others do not respond, we have to unravel and understand this complexity. And therefore, we rely on the tools that allow us to measure this complexity with scale and high resolution. And we from 10x Genomics, with our partner Novogene, we can help you here in providing and tackling with these tools this complexity. How much is actually 40 trillion? So it's just a number that you may have no understanding about, but if you would translate 1 second into the number 1, then 40 trillion seconds would be roughly 1.3 million years. So this is just a crazy amount of number that the human body is made of. And there is enough research for all of us in the next centuries, giving this amount of cells that are there. And we from 10x Genomics, we believe that the complexity of biology can only be understood at the cellular level. And a cell is basically the functional unit of an organism. And we now know that many diseases arise from a cell misbehaving, giving cancer, for example, as a prime example here. That brings me to the research field, a bit of single cell analysis, which is, as a bit surprisingly, actually an old research field. So coming from the first microscopic pictures here in the 700s. But these single cell analysis is primarily based on optical readout and has only limited scale or resolution. If we think of single cell analysis today, we think more of single cell transcriptomics or single cell gene expression, where in RNA gene expression, bulk RNA is the most commonly used method of detecting messenger RNAs. It fails to uncover the cellular variety, which can only be seen at single cell resolution. So if you take this, for example, you would miss that this red gene pattern is basically only expressed in these specific cell types. So you may have seen these t-SNE projection here, which basically is just a way to visualize data and single cell data. So a bulk RNA-seq dataset would just appear at this one-dimensional dot in this 2D t-SNE projection, as it represents the mean gene expression profile of many cells. On the other hand, single cell gene expression would give you these highly granular clusters and groupings of all the cells that you have analyzed, and they are grouped together based on the individual gene expression profile. So each single dot here would represent a single cell. And this projection just groups them together on how similar or different they are to each other. So single cell analysis, single cell gene expression can be also seen as a genetic microscope that allows you to find a needle in a haystack and to define and look at small subtle differences between cells and cell clusters. Another widely stretched analogy of single cell analysis and bulk is this car model here. So you would look at bulk, you would see that it's made of some plastic and metal and maybe leather, but only single cell analysis would give you a complete part list. So you would see the individual part and also would see that rubber is, for example, mostly found in tires and leather mostly in the seats. And as a next step of analysis level, the spatial analysis is coming up to speed, and I've also seen in the first slide from Novogene that this is an offering, but we will focus in this webinar on the single cell analysis part. We from 10x Genomics, we provide you with the complete toolbox of platforms. We call them Chromium, Visium, and Xenium that allow you to analyze your samples at single cell and also at spatial resolution, with single cell being the gold standard for single cell gene expression profile and Visium and Xenium being our spatial solution. So as mentioned, let me focus today on the Chromium part. So Chromium has entered the market in 2016, and since then, this system has catalyzed what is now called the single cell revolution. So coming from just a couple of cells in a single assay at high cost to millions, 100,000 millions to even billions of cells, and up to date, over 15 billion cells have been profiled with our assays, resulting in more than 10,000 publications by our customers and users, mostly of them in high impact publications, and with more coming every day. We also believe that Chromium is the gold standard because we provide the most comprehensive single-cell platform as we enable multi-omic analysis on single-cell level. So we can read out obviously the gene expression profile, but we can also, from the exact same single cells, we have assays that give you cell surface protein information, as Sonny mentioned in the beginning, also immune profiling, so T cell or B cell receptor profiling. We also have an assay that allows chromatin accessibility and so on. We also believe that we have the most comprehensive single cell platform is that we enable a lot of sample types with our Chromium assays. So whole blood was mentioned by Sonny. We also enable frozen tissue, flow sorted cells. Fixed and FFPE tissue is an addition that can also be done with specific assays that we have, and we are also species agnostic with many of our assays. And quite an impressive list, Sonny, by you in the beginning on. I even saw like shark and tiger on the list, so I think maybe some of your single cell users have already used this also with our assays. We offer three major types of single-cell assays, as mentioned, depending on your research goal and your research needs. We call them universal assay, flex assays, or epigenetics or AP assays, where basically FFPE would only be compatible with the flex assays. I will focus today on the universal assay, which is a reverse transcription-based assay. That brings me to the two major principles of how our assays works. We have a let's say traditional reverse transcription-based assays, so oligo to T priming to a poly A tail, and we reverse transcribe every messenger RNA that there is in the single cell. On the other hand, we have a probe-based assay, which is basically like a microarray, but at high scale, and the whole transcriptome is, and that would enable you to work with FFPE samples as well, because it can work with fragmented and truncated RNA species. So how does Chromium and our technology work? A short excursion here on a bit more technical side. So two buzzwords you need to remember when you think of our technology, and this is partitioning and barcoding. So individual cells, represented here by these small white dots, are captured in water and oil droplets in our microfluidics channels, and each of these droplets contain a unique gel bead. And we have, for example, for the 3' assay, we have roughly 3.6 million different and unique gel beads. And once a cell is captured with a gel bead in a water and oil droplet, we call them gems, so gel bead in emulsion. So you see here in this nice animation, or it's not an animation, it's actually a video of our channels, how fast and robust and streamlined this is. So now each one of these gems, so these gel bead in emulsion, acts as an individual reaction chamber. So it is an enclosed environment where we can perform our reaction. So in each of these droplets, the gel bead is then resolved, it releases the enzyme, the cell is lysed, releases all the RNA, and within this droplet, we then reverse transcribe the mRNA, or basically everything that is polyadenylated or captured on our beads. And by that, we give every transcript a unique 10x barcode that has been attached to a gel bead before. So this unique barcode will then give each individual droplet and thus each individual cell a unique address that can be later on identified as a single cell in the sequencing data. Here's a brief overview on the workflow. So you start with your single cell suspension, your partition and barcode. Using our Chromium instrument, you generate a library, you sequence this library, and the sequencing data then is processed via streamlined and algorithms and pipelines we have established, and that's also Novogene is offering. And at the end, it's you have the data. We provide data visualization tool, and also Novogene can help you. So this is basically the wet lab part in the beginning, and after sequencing, we call it dry lab part because you can do it not sitting on a lab bench. A small addition here on sequencing consideration. So obviously our fantastic partner Novogene can help you and give you some estimates. So we do come up with recommendations, but obviously it strongly depends and is influenced by your sequence, by your research goals. So if you just want to profile subpopulations, you may not need to sequence so deep, But if you're looking for really, really rare transcripts, you need to sequence deeper. So just keep in mind there is this kind of saturation curve, so that at one point you don't get a linear correlation of sequencing depth to new genes discovered. But 10x and Novogene, we are here to help you to find the right sweet spot for sequencing depth. As briefly touched in the beginning, we all can also capture proteins, so cell surface proteins in this case. And herefore, we basically use a simple and smart trick that we exchange the fluorophore that is usually coupled to an antibody with an oligo. So we have here oligo-conjugated antibodies. And if you recall that we reverse transcribed everything that is captured in a droplet, in a gem, we also capture these oligo that is attached and dragged with the cell into the droplet. So in parallel to the gene expression profile, we can also read out protein information. And this is especially interesting as when we exchange a fluorophore to an oligo, we are not limited anymore by overlaying spectra of those fluorophores. So with current FACS and flow cytometry, you may, if you're good, can have 6 or 7 antibodies in parallel, but we are not limited anymore by that because we simply have a genomic barcode readout. So you can do tens of hundreds of antibodies in a multiplex fashion on the exact same cell in the same time. So here we work with antibody experts, which are BioLegend, Protein Tech, and Abcam, and they provide pre-mixed, ready-to-use cocktails with over 100 conjugated antibodies available off the shelf. So just click and choose, add it to our assays. Pretty straightforward protocols. Additionally, with our Genomics Universal 5' assay, we can also specifically amplify the VDJ repertoire, so the sequencer from B or T cell receptors, which, for example, would allow you to track clone expansion of specific clonotypes. And it will also give you paired information from the single cell. So for a T cell receptor, you would see which alpha and beta chain can be found together in the single cell and what type of cell is that is. If it's a cytotoxic T cell, a T helper cell, a regulatory T cell. And for B cell receptors, it will give you paired heavy and light chain information. So to briefly summarize, the Genomics Universal Assay gives you a broad range of multi-omic analytes that can help you with your research. How to get started with a Chromium Single Cell experiment? We understand that this might be a bit overwhelming to you, and there are so many things to consider. But you shall not worry. We and also Novogene, we help you to start. We have tons of resources on our website, so on 10xgenomics.com or novogene.com. Just check it out. We have a lot of information there, beginner's guides, blogs, protocols, and so on and so forth. But maybe to guide you a bit more by the hand, you can ask yourself a couple of these questions. So what is your scientific question that you want to answer? How do I prepare and process my sample? Are there maybe some best practices available already? How many cells and replicates do I may need, or do I get? Sometimes the availability of the sample is basically determining a bit what you can do. What is the sequencing depth you need? As I mentioned in the beginning, it depends on if you're looking for rare cell types versus just like subpopulation characterization. And how do I analyze and visualize the data? And I wanted to pick this with these two people here chatting each other. Talk to each other, talk to us, talk to Novogene, talk to data scientists right from the start, so they can already help with experimental planning, setting things up the right way, so you don't end up something with the data and then you realize, oh, I should have included this specific control here. And visit our support hub, 10xgenomics.com/support. There's tons of materials, well prepared depending on experimental design planning, sample preparation, library construction. Click and choose. We have really fantastic material there. It's all 10x validated and official. And reach out anytime to our super cool support team at support@10xgenomics.com, which I guess they have a mandate of answering within 24 hours, so you get an answer back really fast. Here's just an example of over 40 pages cell preparation protocol, so definitely urge you to look into that if you're strongly considering single cell. As a general rule, as you know, in planning these things, 10 minutes before invested saves you 2 hours afterwards. So take your time, consider everything beforehand, as these cells and samples are precious, and we want to make the best out of your samples. We have, as mentioned, just an overview here. I do not go into this into detail, but we have general guidelines, sample-specific guidelines, sample improvements. So tons of material here. And I guess there was a question in the chat beforehand. We will send out the presentation to you afterwards, so you don't need to worry that you might miss something. Yeah, just another example, specific protocols depending on the tissue you're working in, tumor tissue, embryonic or human PPMCs. And this is an overview that also shows you that we have several methods to preserve samples, because we all know it's a pain to work with live cells and they are fresh and you need to run from one floor to the other to get them prepared. So there are certain ways with certain assays to preserve cells, so naming, for example, cryopreservation or mild fixation with DSP methanol that is compatible with the Genomics universal assays that I have briefly touched here. Formaldehyde, which is a strong fixative, is only compatible with the probe-based flux assay, and 4-degree short-term storage would also be compatible with all of our assays. So now I have touched a bit and hopefully educated a bit on how we and Novogene support you on the wet lab part, so from sample prep, on partitioning and barcoding. Hope you remember these two buzzwords, to library construction, to also sequencing. But what about the rest? What about the dry lab part, the data processing part? And with that, I hand over to my dear colleague, Martin. So I need to stop sharing my screen. I hope Martin has logged on already. Alrighty, here you go. So many thanks.
Yep.
And I will be quiet now and hand over to Martin.
And I will share my screen. I hope you can see my screen and hear me.
Yes.
The full screen. Okay, super. So thank you, Nico. Thank you very much. And thanks all joining us here today. And thanks also Novogene for inviting us for this seminar or webinar today. I would like to take the next couple of minutes to show you a little bit on how the data analysis part looks like. It is, in fact, a road which is not straight, but sometimes it's a windy road. And Nico already mentioned that a lot of stuff can be already be discussed in the experimental design part, and it's very helpful. And I think it can spare you more than 2 hours. Sometimes it can spare you days if you do it correctly in the beginning. Then he has talked about the lab workflow, which is really supported by a lot of protocols and background material, and of course the expertise of our service partner here. And then we get to raw data processing once the sequencing is done, because we want to transform the sequencing data into quantitative biological information. We do some quality controls, and the visualization and interpretation part is what I would like to cover here today. So how does a data analysis workflow look like? It starts with the FASTQ files coming from the sequencing. We do the read, processing, and quantification. We do some quality filtering. We do data integration, clustering, because we want to know which cells actually have similar expression profiles, for example, on that. And by that, get also an idea of the composition of our tissues and what is happening there, especially then as well in getting the right cell types annotated. And last but not least, also, of course, differential gene expression analysis to decide what might be different in diseased cell situations to normal cell situations. And of course, a lot more. Nico talked about VDJ, so also looking into the makeup of or the composition of immune cells on a molecular level. The one aspect I would like to mention here is that, again, here, this is not always a straight line. It goes in circles, and you need some tools which allow you to address it in a very smooth and easy way. Because if you do, for example, a quality filtering, you will start again here and have a different data set. If it's filtered than the original data set, you can do it with different stringencies, etc. Same holds true for clustering, cluster sizes, etc. So just bear in mind that this is actually probably the cheapest part of your analysis, because this is only taking your time and some compute environment, but you can squeeze out a lot of data and a lot of results in here if you really mine yourself a little bit deeper into the data sets. It's relatively easy because we have a lot of tools at hand. And the first part, actually turning the sequencing data into meaningful biological entities and assigning it to cells and so on, is done by Cell Ranger. Can be provided also by the service provider or within our cloud, or you do an on-site installation of that. Secondly, what I will touch today is the Loop browser, a very intuitive browser where you can take the data directly after the Cell Ranger analysis in and have some visualization without needs of being able to do command line programming or stuff like that. You can just directly dive into that. And last but not least, of course, there are a lot of community developed tools which allow you and your bioinformatics partners to mine really deep into the data. And this is an ever evolving community. And at the moment, we are counting almost 2,000 different programs which allow you to mine deeper into the data. Cell Ranger has had some additions, especially in the last year. It is now allowing you also, since Cell Ranger 9, currently we have Cell Ranger 10, to do also high-level cell typing. So directly the results after the Cell Ranger run will contain also information about the cell types, especially if you are sticking to the human genome and human samples. It brings out the data in a way that you can do instant visualization in the Loop browser. And I just would like to mention that the cell type annotation is based on cell by gene database. And we, together with some partners, have developed this and bring it into your hands immediately after the analysis is performed. Once you have done that, you get your data and you can directly mine the data with the Loop browser. And the required skills is you need to install it on a Linux or a Windows machine. You should have an idea about your experimental design. I will show you how you can then look into also differential gene expression and stuff like that. So it's easy, straightforward. By the way, it's all free of charge. You can download these programs directly from our homepage, from our webpage, and install it on your computer without having to pay anything for that. So that is provided directly by 10x. I would like to show you an example how a data analysis can look like. It is also a data set where we have a tutorial for, and you can download it also directly from our webpage. It consists of 21 flash-frozen tissues from treated and untreated non-small cell lung cancer samples. To be precise, it's lung squamous cell carcinoma. And additionally to that, normal adjacent samples are also in here. And the data was in principle obtained by doing snap freezing of human lung tissue, fixation, then dissociation, single cell suspension, and then it went on the Chromium X. And you can see this is quite a big data set, which you then directly can have a look at. As said, you can download this directly from our webpage. And also what I'm showing you here is explained in detail also within this tutorial, which is also can be found via this link here. So first step, Cell Ranger analysis of the data. Fast queues go in. They are aligned. We aligned the sequence reads to a reference, in this case, the human reference. We get a BAM file. We do counts, count the number of alignments to features of interest. We call the cells by using the 10x barcode, which Nico was showing you. And also Cell Ranger already performs a first analysis layer. It produces t-SNE and UMAP projections. It does clustering, and it does also some differential analysis between the called clusters. Once you have done that, It will all be visible in a folder called outs, and you can see that I highlighted a couple of those files here. It's the sample C-loop public domain data format, actually, where the data model can be used also in a lot of community developed tools. So just would like to highlight that we always stick to public domain formats for compatibility with what is out there. So it's not a closed system, it's a really open system. And last but not least, we have a web summary giving us some data assessment of the experiment. If you click at the web summary, you will learn things like how many cells are in a specific subset of the data, the number of reads, the median genes per cell, the total genes detected, the medium unique molecular identifier by the cell. That is actually for quantitative aspects a relevant factor, and the number of confidentially mapped reads in the cell. You get the first clusters. So this is a t-SNE projection, and you can see the density on the one side, and on the right-hand side, you can see the different clusters, which are probably reflecting different cell types, but are in this case just a graph-based clustering. But you can see that this is nicely spread out here. And as Nico already mentioned, each of the dots is a single cell. And the closer these dots are together, the more similar they are in their expression profile. And the expression profile of the clusters actually is also already provided after the cell range analysis. So each cluster is calculated, there is calculated the expression intensity of each gene in here, and we calculate the actual expression level of the gene in cluster number 1, for example, against the base mean of the other clusters, and you can switch and choose directly which would you like to sort for, whether it's cluster one or one of the others. And a p-value is also provided for giving the significance value. And with that, I would like to show you that you just open the C-loop file, and that's what I've done here already of the data set. We have in total 157,000 and something cells in here. And if I open that, you will just see how the program looks like. You can see that we have a t-SNE clustering here. You can see all the clusters. You can switch the clusters, and you will see also down here always the expression values of the different clusters. And if you switch them on all again, you get the expression value I showed you on the table before. You could also use a UMAP clustering instead of the t-SNE. Then it's a bit more spread out, and you can see how this looks like. Again, you can switch the different clusters, or you can add also different additional information like disease states, as we have it here, untreated, treated, and normal adjacent. And from one single UMAP, we can spread it out into separated UMAPs directly here, as you can see it. And we can switch on the cell types, and you can see the different cell types. For example, here, these would be the basal cells, the subset of that, in the different disease conditions, untreated, treated, and normal adjacent. Let's go back to the disease state. One thing you would be probably interested in, what is different between the different disease states, and we can just run a differential expression analysis by clicking the blue button down here against the entire data set and start the analysis. It will take a little bit, but what we get now very easily by clicking on that here is a calculation of differentially expressed genes between the different disease states. So it's just one click away. And here we go. We get a table of that, and the top scoring in untreated cancer is carotene 14, for example, with a very low p-value, so this is very significant against treated and normal adjacent. We can switch here to all detected genes so that we can sort up and down regulated ones, as you can see it here, and we could sort also for the treated cancer and the normal adjacent. Let's go back to the untreated one. We have this table here, keratin 14, keratin 5, etc. And we can easily export this to a CSV now. And that's how it looks like. And you can open it simply with Excel. And here we go. And you have the whole analysis here. And we could take the first 30 now of those being upregulated in untreated cancer, take them and take a third-party tool like Enricher, which is giving us an enrichment analysis. We just paste the 30 entities here, 30 genes, submit. And if we go to cell types and look at the cell marker 2024, we see that basal cell lung human is the top scoring category for those being differentially upregulated here with the marker genes. Very quick analysis. I don't want to save that, but you can see this is here coming up. It's in basal cells, basically. So we can go to our cell types and have a look how the basal cells look like. Let's switch them all off and just the basal cells. And you can see in untreated cancer, treated cancer, we have a bit more than the normal adjacent tissue already. And if we want to know where keratin 40 now is really expressed, we just type it in features and see the intensity of the gene expression of keratin 14 in the different subsets. And it's strongly expressed in untreated cancer, very well in line with what we just saw. And this is, by the way, the same region as we saw before for the B cells. So all makes sense and comes nicely together. So what I wanted to show you here is how quickly you can do a differential expression analysis. You don't need to go to the command line to get that. Cell Ranger directly provides you with the CLOOP file. You can open that. Of course, you could do a lot of different analysis. We could subset now, for example, to only looking at the basal cells, have a look what else is expressed in there. And we even could do a pseudobulk analysis, doing analysis for the different replicates which went into this analysis and see what's coming out from there. All is possible directly with this graphical user interface, so a very straightforward way. Coming back to keratin 40 for a second, by the way, it's just also something which is known from literature already. Keratin 40, high subpopulation mediates lung cancer metastasis, potentially through a specific gene upregulation of GKN1. So it is a relevant finding. I'm not saying that I provide you new insights. I just wanted to show you how easy you can get your data analyzed into a really meaningful biological direction. With that, I would just like to mention that, of course, you can do way more with the Loop browser. You can do a lot of comparisons between groups and clusters. You can do cluster annotation. You can subset your cells in here very easily. And you can restructure and substructure also very quickly here. So get a lot of heat maps, clonotype analysis, spatial information can all go into that. And last but not least, I would like to mention that the data set I was showing you is not the only one we have here. We have for Chromium Single Cell, we have more than 500, but we also have for Visium and for Xenium, we have a lot of data sets being available directly for download, everything free of charge. In addition to the programs, as I just mentioned, Loop browser and Cell Ranger are also provided free of charge. With that, I'm at the end of my presentation. I would like to thank you. Also showing you a picture of a subset of the company with our last meeting in California. And I hand over back to Novogene with that. And I'm happy to take, or we are happy to take questions. And I stop sharing my screen, I guess.
Okay. Thank you, Martin and Nico. We have quite a few questions that are coming in for both of you. So the first question we have is, just a moment, I need to be able to see my screen.
All good.
All right, I can see it now. So the first question we have is from Maria asking, how reliable is single cell RNA sequencing of FFPE tissues and also single nuclei RNA sequencing of FFPE tissues? Do they really contain all the mRNAs in the FFPE samples?
Yeah, I can take this one. Thank you, Maria, for the question. As you remember, we have this probe-based assay for FFPE because obviously it's fixed and it's truncated, and we cannot rely on the poly-A-tail for reverse transcription. And what we see is that is highly reliable on getting basically the information that you would also get from freshly prepared samples. Because when a transcript is there, these probes that basically bind to these region, and we sequence these hybridized products. We make sure to get all of these present transcripts by tiling probes over the span of a transcript. So if we have for 95% of all the genes, not only one probe pair, but three probe pairs. So not, let's say, in the start of the transcript, middle of the transcript, and the end of the transcript, also having certain splicing junction. So if a transcript is there in the FFPE sample, there's a really, really high chance that it can pick it up with our flex assay. So, and also just to remind you, we have a whole transcriptome probe set against humans, so all annotated human genes for this assay, and a whole transcriptome probe set for mouse. So that means that this assay is not species agnostic, so it's species specific. So you can at the moment only do human and mouse FP samples. Hope
And
that.
if I just may add one thing on that, you can customize probes also for if you would like to have, for example, probes against a specific virus or something like that. We have a protocol available how you would customize the probe sets for that and can integrate it directly into the analysis.
Okay. Yeah, thank you both. Looks like there is always a solution, no matter your sample type. The second question, I guess, would be for Martin. The question is, does Loop browser support non-model species that have a reference genome, for example, domestic cat?
You can integrate that into the Loop browser analysis. That is not an issue.
May I
So you
ask?
need a
Yeah, go ahead. Sorry, go ahead.
So what you need is you need a reference, and you need the reference indeed also already on the cell ranger level. If you have a non-model species as we have them for human mouse and so on, you can create your own reference which contains the backbone sequence of the target organism and the transcript annotation. And if you have that, Cell Ranger provides a command which is called make ref, and you can create your own reference. And if you run Cell Ranger with that reference, again, it will create a Loop browser file which can be directly loaded into Loop for subsequent analysis.
Okay, thank you. The next question is from Tian. She's planning to do single cell RNA sequencing and single nuclei ATAC-seq on the same samples from zebrafish. Her question is basically, should she do multi-omic ATAC, or should she do single cell RNA sequencing and single cell ATAC separately?
May I take this question? Hi,
Yes,
hi,
please.
Tia. Yeah, that's it depends on a variety of things. Obviously, it's the most diplomatic answer I know. But let's say in a commercial sense, it might make sense to go with Multiome right away because it is if you combine it, a bit more cost effective if you do it all in one assay and you only need to do sample preparation once. And at the end, you have truly direct link of the exact same cells of attack profile and gene expression profile. If you do them separately, you need to interfere. It's not the same batch, not the same sample, not the same individual you're using. So let's say, taking from me as a commercial person, I would suggest to go with the multi-ohm combined kit.
And as the not so commercial person, I would probably do the same. I've done a little bit of epigenetics in my life before, so it is indeed very helpful if you have it in one assay together. It helps a lot.
Is there any difference in the data quality when one does them separately versus together? As a follow-up question.
The point is, if you do it as multi-ohm, you probably need to go for nuclei, and if you do it separately, you could do it on the single cell level still.
Yeah. Okay.
Yeah.
Thank you. We have one more question from Olga. She is asking if it is possible to do 10x genomics single cell RNA sequencing on ovarian tissues, because she's asking this because the oocytes, they can be large. Would you then recommend single nuclei RNA sequencing in this case?
Yeah. So as you may remember, when I showed the video of how the droplets and gems are formed, so the microfluidics have a certain physical limit on how big the cells can be, which is 20, 30 nanometers in cell size, if I remember correctly. So the regular cells work, but if the cells are too big, so cardiomyocytes, hepatocytes, and oocytes,
Yeah.
the only way to get single cell information is to get single nuclei information.
Yeah. So basically, if your cells are larger than 30 micron, this is where you would recommend them to go
Yeah,
for
exactly.
single
No,
nuclei
30
RNA
microns.
sequencing. Yeah.
Sorry, it's
Thanks.
one scale apart.
No worries. One last question because we are running out of time. This is a bit different than what you have talked so far. The question is, can one use single cell RNA sequencing to study interactions in bacterial community?
Oh, wow. Um, I mean, the thing is, we capture with the reverse transcript assay only polyadenylated species, and usually RNA bacteria don't have poly A tails. However, there is a way with the capture approach, so this template switching at the 5' end, to also capture non-polyadenylated sequences. So there are certain papers out there which have co-detected viral genes. So if you remember all the COVID studies there. So you
And
can,
also,
there are ways to add bridge oligos to have them also attached to our gel beads and to the barcodes. I
and in
think Martin.
principle, you can use also the Flex protocol to design probes for bacterial sequences. But I would be always a bit careful with bacteria, to be honest, because bacteria, depending on which type they are, they have cell walls, they are sometimes relatively stable. So permeabilization stuff and so on can or will definitely need some protocol modifications.
Okay, thank you. Maybe one last question from our side. We have a lot of researchers that are interested in studying or profiling different cell types in blood. And the question is, should one freeze the blood samples, use them as is, fix it? What would be the ideal strategy, let's say, for patient samples? And is there a difference in terms of number of genes one recovers, the types of cells they profile for blood, depending on the assay they choose?
Maybe I can take this on because one of my customers are the big pharma companies in Switzerland and Germany. So obviously this is of high interest to them. So the best way, I think, and what many of my customers think, is to do cryopreservation of whole blood. But we know that comes with certain technical equipment, with certain costs, with certain logistic things to consider, and with certain trainings of people on site. So cryopreservation is, let's say, the most gentle way, and it also keeps the cells intact, and would also then later on would allow you to analyze TCR and BCR information if that is of interest. The other way is to fix whole blood. We have a protocol there available as well from 10x Genomics, but obviously fixing would then result in streamlining only to go with the flex assay, so you would lose VDJ information. It is a bit more streamlined on site collection side. However, you still need to work with PFA, with glycerol, and that could also be a challenge for some clinical collection sites. So the best way is to preserve the sample at the collection spot. And I think cryopreservation or fixation, which is flex, would be the two best options that are currently available.
All right. And is there a difference in terms of number of genes, let's say, whether one uses flex versus single cell RNA sequencing?
I cannot recall like a direct comparison from the top of my head, but we do
No
see
worries.
no real changes or nothing that this assay we have to live with 50% less genes. So it is same but different.
Comparable. Okay. Thank you. We have a few more questions, but unfortunately, we do not have time to answer them live. We will get to them. We will send you an email with answers to your questions. And thank you, Martin and Nico, for the really helpful seminar. At least I found it really helpful. In the poll that we took, 50% of you said that you struggle with data analysis and figuring out the right technology to get started with the single cell RNA sequencing experiments. So I hope you feel better about this after this seminar. For now, I thank everyone for your time today. And here is a QR code that you can sign up for the 10x Genomics newsletter. You can keep yourself up to date with the latest developments from them, the expert tips, also research applications. We will email you a link to the recording as well as the slide deck. When the webinar finishes, a survey will pop up, and we'll appreciate it if you took a few minutes to complete this, because this helps us organize more relevant future webinars for you. As a token of our appreciation for your attendance at this webinar, Novogene is pleased to offer you a 15% discount on your upcoming project. An email will be sent to you within the next few days with a code that you can then use to apply the discount. There will also be a link in the follow-up email for you to view your certificate of attendance. If you need one, this can be printed from your browser window. In the meantime, feel free to follow us on our social media channels such as LinkedIn to keep yourself updated with our upcoming webinars, promotions, in-person events that might be happening near you. In our next webinar, Dr. Maria Hernandez Soriano will discuss about decoding wheat microbe interactions using shotgun genomics. We look forward to seeing you all there, and thank you again for attending, and thank you for your time. Have a great day.
Thank you as well.
Thank you.
Have a great day. Bye-bye.